The last chapter in the cardiovascular system is Chapter 13, Blood Vessels and Circulation. So in this lecture, we'll distinguish among the types of blood vessels based on their structure and their function. We'll explain the mechanisms that regulate blood flow through blood vessels and discuss the mechanisms that regulate movement of fluids between capillaries and interstitial spaces. We'll describe the control mechanisms that interact to regulate blood flow and pressure in tissues, and explain how activities of the cardiac, vasomotor, and respiratory centers are coordinated to control blood flow through tissues. We'll explain the cardiovascular system's homeostatic response to exercise and to hemorrhaging. We'll describe three general functional patterns in the pulmonary and systemic circuits. We'll identify the major arteries and veins of the pulmonary circuit. We'll then identify the major arteries and veins of the systemic circuit. We'll be identifying the differences between fetal and adult circulation patterns and describing the changes in the patterns of blood flow that occur at birth to conclude this lecture. So this final chapter in the cardiovascular system describes the vessels that transport blood. Blood leaves the heart by way of the pulmonary trunk and the aorta. These vessels branch repeatedly, forming the major arteries that distribute blood to the body. Further branching occurs, creating tiny arterioles, which provide blood to capillaries. Capillaries form extensive branching networks, where chemical and gaseous exchanges occur between the interstitial fluid and the blood. Blood flowing out of the capillary network enters venules, which merge to form veins that deliver the blood back to the heart. The walls of arteries and veins contain three distinct layers. The tunica intima, or tunica interna, has endothelial lining and elastic connective tissue. The tunica media in the middle has smooth muscle with collagen and elastin fibers. This zone, this tunica media, controls the diameter of the vessel, and it is under control of the sympathetic division of the autonomic nervous system, which can result in either vasoconstriction or vasodilation. Last, the tunica externica is the sheath of connective tissue which can anchor the vessel to other tissues. Note the difference in the artery and the vein wall. We have a thickness characteristic of arteries, and this allows arteries to resist the pressure that's generated by the heart on the blood. In traveling from the heart to the capillaries, blood passes through three different types of arteries. Elastic arteries are the first type of arteries that leave the heart. A couple of examples are the pulmonary trunk and the aorta. They have more elastic fibers than smooth muscle because they have to absorb the pressure changes readily, stretching during the systole and preventing very high pressure. They're then relaxed during the diastole, which prevent very low pressure. In muscular arteries, the tunica media contains a high proportion of smooth muscle and little elastic fiber. Examples are the carotid arteries. And finally, in arterioles, the tunica media has only 1 to 2 layers of smooth muscle. And this allows it the ability to change diameter to control blood pressure and flow. Capillaries have a tunica internica only. Essentially, they're just endothelial cells with a basement membrane. And this forms an ideal mechanism for diffusion between the plasma and the interstitial fluid. These thin walls provide a short diffusion distance, and the small diameter slows flow to increase the diffusion rate and provides a huge surface area, again, to increase diffusion. This figure is showing the detail of how arterioles and venules form capillary beds, an interconnected network of capillaries. The entrance to a bed is regulated by precapillary sphincters, which is a band of smooth muscle. Relaxation of the sphincter allows for increased flow. Constriction of the sphincter decreases flow. This occurs cyclically and is referred to vasomotion. Control is local through autoregulation. Back to this figure again, the veins then collect blood from tissues and organs and return it to the heart. The venules are the smallest of these structures. Some venules are so small that they lack the tunica media. These are drained by medium-sized veins. In medium-sized veins, the tunica media has several smooth muscle layers. These are then drained by large veins. Large veins have a thin tunica media and thick collagenous tunica externa. These large veins have thinner walls than arteries because there's relatively low pressure in the veins. In limbs, veins contain valves to prevent backflow of blood towards the distal ends, and they increase venous return. As long as the valves function properly, any body movement that compresses a vein will push the blood towards the heart. And so we've reached a checkpoint where you can test your knowledge so far. The purpose of the cardiovascular system is to provide all the tissues of your body with fresh oxygen and nutrients. The only way to do that is by a constant flow of blood. Normally, blood flow is equal to cardiac output, meaning that an increased cardiac output leads to an increased flow through capillaries, and decreased cardiac output leads to reduced flow. Blood flow through capillaries depends on pressure and resistance. These two factors have opposing effects, meaning that increased pressure increases flow, decreased pressure results in decreased flow. Increased resistance decreases flow, and decreased resistance leads to an increased flow. So to maintain blood flow, the heart must generate enough pressure to overcome resistance in the pulmonary and systemic circuits. So when liquids like blood are compressed, like under a ventricular contraction, their pressure increases. This pressure is called hydrostatic pressure. When there's a pressure gradient, so an area of high and an area of low pressure, the high pressure liquid will flow towards an area of low pressure. Therefore, flow rate is proportional to the pressure difference. The hydrostatic pressure in the cardiovascular system is called circulatory pressure. And circulatory pressure is responsible for the force of blood through the arterioles and into the capillaries. Circulatory pressure is divided into three components: arterial pressure, measured as blood pressure, capillary pressure, and venous pressure. For every force, there's an equal and opposing force, a little physics for you, and the force that opposes movement is called resistance. In the cardiovascular system, resistance opposes blood flow, and therefore circulatory pressure must be high enough to overcome total periphery resistance, which is the resistance of the entire cardiovascular system. Highest pressure gradients exist in the arterial system because the high resistance of the arterioles is called peripheral resistance. Sources of peripheral resistance include vascular resistance, which is caused by friction between blood and vessel walls. The amount of friction is due to the length and the diameter of the vessel. So the longer the vessel, the higher the resistance. The smaller the diameter, the greater the resistance. Another factor Another source of peripheral resistance is viscosity. So viscosity is due to interactions between molecules and suspended materials in a liquid. So low viscosity fluids flow at low pressures. High viscosity fluids flow only under high pressures. And the last is turbulence. So eddies and swirls in fluid flow is turbulence. In smooth-walled vessels, the turbulence is very low. Slow flow near the walls, faster flow in the center. Injured or diseased vessels or heart valves can increase turbulence and decrease flow. Of these three sources of resistance that I listed, vascular resistance, viscosity, and turbulence, of these three sources, only vascular resistance can be adjusted by the nervous or endocrine system to regulate blood flow and, therefore, blood pressure. I mentioned the pressure gradient that exists in the cardiovascular system, with the highest pressure in the arterial network, but this pressure fluctuates. It rises during ventricular systole, called systolic pressure, and it falls during ventricular diastole, so diastolic pressure. So when measuring blood pressure, we record systolic over the diastolic pressures. And this can be noted by a pulse, the rhythmic pressure oscillations that accompanies each heartbeat. The differences between the systolic and the diastolic pressures is the pulse pressure, which lessens as the distance from the heart increases. This figure depicts the pressure within the systemic circuit. Systemic pressures are high in the aorta, peaking at around 120 millimeters of mercury, and the lowest is at the vena cava, averaging just 2 millimeters of mercury. Systemic pressures are highest at the aorta and lowest at the vena cava, like I just said. Now, notice the oscillations, the up and the down. This is the pulse of the ventricular systoles and diastoles in the atrial network. By the time the blood reaches the capillaries, no oscillations can be detected anymore, and the pressure at the capillaries drops from 35 to 18 millimeters of mercury. The pressure of blood in the capillary bed, the capillary pressure, pushes against capillary walls just like it does in the arteries. But unlike the arteries, thin capillary walls are permeable to ions, nutrients, wastes, gases, and water. Capillary pressures cause filtration out of the bloodstream and into the tissues. Most materials are reabsorbed into the blood, but some materials are picked up by the lymphatic vessels. And we'll discuss the lymphatic system in the next chapter. This continuous movement and exchange of water and solutes plays an important role in homeostasis because it maintains a constant communication between the plasma and the interstitial fluids. It speeds up the distribution of nutrients and hormones and gases to the tissues. It also assists in the movement of insoluble molecules and flushes bacterial toxins and other chemicals to the lymphatic tissues straight to the immune system. This figure is depicting how materials move across capillary walls by diffusion, filtration, and osmosis. So you can see capillary hydrostatic pressure, the CHP, is high at the atriolar end. So this will filter water out of the capillary. Solutes diffuse across this capillary lining, driven by their individual concentration gradients. Water-soluble solutes will squeeze between capillary cells. Lipid solutes diffuse through the cell membranes. Large solutes like plasma proteins are too big to diffuse, so they're important in maintaining the blood osmotic pressure, the BOP. And the blood osmotic pressure is higher than the interstitial fluid. So as the capillary hydrostatic pressure drops over the length of the capillary, the blood osmotic pressure remains the same, so it favors reabsorption of water in the end. The difference in pressure in the venous system is very low compared to the arterial side, from 18 millimeters of mercury in the venules to 2 millimeters of mercury in the vena cava, which means that only 16 millimeters of mercury is pushing blood in the veins. Fortunately, as blood travels through the venous system, the veins become larger as they get closer to the heart. So the larger the diameter, the less resistance. But 60 millimeters of mercury is not enough to overcome the force of gravity, especially when you're in a vertical position. So two factors help to propel venous blood towards the heart when you're not lying down. That's the muscular compression, which anytime you have movement of the skeletal muscle, it'll press on the outside of the veins and it will move the blood in the veins. And remember that we have venous valves, so that movement is in a one-way direction, always back towards the heart. We also have the respiratory pump that is a result of thoracic pressures every time you inhale and create pressure. So we've reached the last checkpoint of this recording.